Hello AP Biology students, we're currently in Unit 4 talking about the cell cycle, and I'm going to spend some time reviewing the phases of mitosis. We're not going to go over any specific section, but we're going to just review what mitosis looks like at a cellular level. I'm going to start off by showing this easy diagram of what mitosis looks like, where we start out with a cell that's diploid, and for humans, we have 46 chromosomes in that cell. Our end goal is to make 2 new cells that are going to have each 46 chromosomes. These cells are also going to be diploid. The reason why the chromosome number doesn't change is because we're creating body cells or somatic cells, and these cells want to have the same exact genetic information as the parent cell. When talking about the amount of DNA, it's important to realize that we do double the amount of DNA during interphase, specifically in S phase. You can see in interphase, the cell is going to grow in size and also double the amount of DNA it has in its nucleus. What will happen during mitosis is at one point it's going to splitting the DNA in two. But that takes us back to the original amount of DNA, which is what we want because we want 2 new cells with the identical amount of DNA. Let's go over each of these steps and what they look like of the cell cycle, focusing in on mitosis. Remember that the cell cycle is made up of interphase and M phase, which stands for the mitotic phase or mitosis. During interphase, the chromosomes aren't going to be condensed. You can see in interphase, again, the chromosomes haven't condensed yet and we still have a nucleus. During prophase, you can see the chromosomes have condensed and we're starting to get ready for cell division. During this phase, the nucleus will also start to break down. Now, this diagram doesn't have prometaphase because some diagrams just leave it out, but this is when the spindle fibers are going to attach to the kinetochores of the chromosomes. What they do after they attach is move them to the center, which is called metaphase. During anaphase, the spindle fibers are going to shorten, which rips the chromosomes apart. Each one of the chromosomes will now only be made of 1 chromatid. But basically, we are making sure that each new cell will only have half of the double genetic information. In telophase, you can see the nucleus is starting to reform and the DNA is going to also start to uncondense. During cytokinesis, the cell will finally split, creating 2 new cells. Here's another way of looking at it. You can see interphase, where the chromosomes haven't condensed. Prophase, the chromosomes have condensed, the nucleus starts to break down. Metaphase, they start to align in the middle. Anaphase, they start to pull apart. Telophase is when the chromosomes start to uncondense and 2 new nuclei start to form. And cytokinesis is the splitting of the cytoplasm. I won't take you through every step, but I want to show you that this diagram includes prometaphase, where you can see the chromosomes being attached to the spindle fibers at their center or their centromere, specifically at the kinetochore. So not every diagram will have a prometaphase, but some will because they want to show that connection between the spindle fibers and the chromosomes. I want to end here by looking at some actual cells and what they look like, because you also need to be able to identify what the cells look like in a microscope. You can see at the top, the interphase is a dark nucleus where the chromosomes haven't condensed. Prophase is when the chromosomes start to condense. They look a little bit different than the diagrams. You can see again, they have those long chromatids, but they are condensed. Metaphase, they align in the middle. Anaphase, they start to pull apart. One thing I want to mention here is it's hard to tell when anaphase stops and telophase begins because this is an ongoing process. It's not like a start and stop motion. This continuously goes throughout the whole M phase. But somewhere around this point, we have telophase where the 2 new cells start to become evident. And then here is cytokinesis where the cell starts to split. Again, it's important for you to be able to identify these cell phases, whether it be a diagram or real cells.